welcome to our newest episode of the LilBUBome video log. In today's episode, I'll talk about what exactly it means to sequence a genome and how we went about sequencing bubs. So the starting point for our story is that we know that the instructions for how to make a cat are encoded in the genome of a cat. And this genome is essentially one big gigantic word of 3 billion letters of DNA. And we call these letters bases, and they can either be A, C, G, or T. And when we say that we want to sequence a genome, what we really mean is that we want to determine the order or the sequence of these individual letters. So how exactly do we go about to obtain such a sequence? So for us, the starting point was a blood sample of bubs, which got shipped all the way from Indiana to Germany. And then Daniel and Dario extracted DNA from this sample. But in itself, this DNA is not yet ready for sequencing. There are still a couple of details that we have to consider. One of these is that 3 billion letters is actually a really long word. And even though there are multiple technologies out there for sequencing, currently none of them are able to sequence the entire genome from beginning to end. So what we do is we apply a kind of workaround where we cut the genome up into smaller pieces, which then become much more manageable. In the case of Bub's genome, we made two separate preparations of her DNA. One where we cut it into small fragments that were 300 bases long, and one where we cut it into small fragments that were 500 bases long. Now, once you've obtained these smaller fragments, there's a couple of things you can do. First of all, instead of having to sequence the entire 3 billion letters starting at one end and slowly proceeding all the way to the other end, you can sequence each fragment simultaneously. And by sequencing the fragments in parallel, the entire sequencing process becomes much faster. Another thing that you can do is instead of sequencing each fragment only from one end, you can at the same time also sequence the other end of the fragment. And the so-called paired end sequencing gives you much more precision. Speaking of precision, there's one more thing that we have to keep in mind, and that is that none of the methods at our disposal are 100% accurate. So it can happen that during sequencing we actually read an incorrect letter, as you can see, for example, here in the 5th position where we're reading an A, even though the correct letter would be a G. So one way around that is that instead of sequencing every fragment only once, we can sequence it many, many times, and then look what the majority of the reads tells us. And here in the 5th position, the majority of the reads is of course a G and not an A, so that tells us that the correct letter would be a G. And we call this sequencing many, many times the depth of the sequencing. In the case of Bub's genome, it was 40x, which means that every single fragment was on average sequenced 40 times. Okay, having considered all these details, having cut Bub's DNA into smaller pieces, and having prepared it in a way so we could sequence from both ends many times over, the sample is now ready to be loaded onto a sequencer. And for the tech aficionados amongst you, what you saw in the previous pictures was an XSeq 500 machine, which was the one that we used for the sequencing. And that's it for today's episode, but stay tuned for the next one. Love the mambo tonight.